Hello, everyone. My name is Aman Chauhan. I'm a medical oncologist at University of Miami. I lead our Neuroendocrine cancer program at Sylvester Comprehensive Cancer Center. Thank you, LACNets, for giving us opportunity to discuss this important randomized phase 2 multicenter study sponsored by NCI. Today, we will be discussing ETCTN 10558. If you remember, we discussed the phase 1 ETCTN 1038 previously, which was the building block for this phase 2 study. I'm happy to notify our audience that the study ETCTN 10338, which was phase 1, was successfully completed, and we are now into the phase 2, which we'll be talking about today. So why do we need to conduct this study? You are aware of a very important drug called Lutathera in our space has been a significant game changer for our neuroendocrine tumor patients, the well-differentiated gastroenteropancreatic neuroendocrine tumor patients. This drug was studied in an important NETTER-1 study that you are aware of, and these are the data from NETTER-1 presented by Dr. Strasberg, showing how well Lutathera works as compared to the control arm, the top line in the green showing significant improvement in progression-free survival as compared to the control arm. This study led to FDA approval of Lutathera in the US, and we no longer had to send patients abroad to get PRRT. And again, a huge milestone for a neuroendocrine tumor drug development in the US. So PRRT now is mainstream, and a lot of patients, hundreds of patients, get PRRT or Lutathera in the US currently. Having said that, there is more room for improvement. Currently, Lutathera response rate, that means number of patients where Lutathera is able to shrink the disease, that is in the range of 14 to 17%. We would like to see if we can enhance the effectiveness of Lutathera so that our patients with large, bulky disease, they can show more drastic shrinkages. There is a significant improvement in median progression-free survival with Lutathera, but can we do better? Can we make it even double? So again, we are always greedy and we want better for our patients. So we want more and further refinement and improvement in the treatments. So with that aim, we designed the study, adding a radiation sensitizer to Lutathera, where Lutathera still does the most heavy lifting, is the main stay of the treatment or the backbone of the treatment. However, the goal is, by adding another drug to Lutathera, can we improve the effectiveness of Lutathera and the kill rate of Lutathera for the cancer cells? We stumbled across a drug called Triapine, and this is a ribonuclide reductase inhibitor. I won't go too much into the science of it. However, just to simplify the concept behind how this drug works, Lutathera is a DNA damaging agent. Once we inject this drug, it goes and circulates in your blood and latches on to the somatostatin receptor positive tumors, and then radiates these neuroendocrine tumor cells. Radiation causes DNA damage, and without a functioning DNA, the tumor cells should die. However, you would think that all these cells would die, but in reality, the tumor starts growing again after several months or years. And one of the reasons why that happens is the tumor cells are smart and they have found ways to repair their DNA. This particular drug works in a way to slow down or stop that repair process. So it makes sense adding this drug to a DNA damage agent, where Lutathera does DNA damage and Triapine slows the DNA repair in the tumor cells, and together they would synergistically. We tested this hypothesis in preclinical models. We actually just published this paper, which describes our work for past several years in different neuroendocrine tumor models, where we tested Triapine with radiation And what you see here is when we add triapine to radiation in our pancreatic neuroendocrine tumor models, significant growth restriction was seen. This was very encouraging and led us to do the phase 1 study. We wanted to test, can we safely test triapine with Lutathera without causing significant harm? And there is a dedicated video about this on the LACNet's website. And I'm happy to announce that the study, which was a multi-center study, enrolled patients through several sites, was a resounding success. It met its endpoint. It was able to accrue patients fast. And what we found, the primary endpoint was to find what is that safe dose of triapine that we can combine with Lutathera. And we were able to find that last year. So it was works spanning across several years, from our lab to phase 1. And we presented our phase 1 prelim data last year at AACR and various other conferences. And we were able to find what is that dose of triapine, and that turned out to be 150 milligram in combination with Lutathera. Now, the next step for this combination is, if it is more effective as compared to standard of care Lutathera alone. So we designed a study that me and my mentor, Dr. Lowell Anthony, are co-leading nationally. And I received an NCI CTEP Career Development Award for this concept. So it's randomizing neuroendocrine tumor patients who would qualify for PRRT. That means should have a positive DOTA-TATE PET scan and had progressed on at least one line of treatment. Into two arms. It's equal randomization, so 1-to-1 randomization between combination Lutathera plus triapine versus Lutathera. So both these patients will get a very good standard of care drug that's called Lutathera, and half of the patient will get on top of that the experimental drug triapine. This study is designed to help us understand whether this combination will improve the response rates. That means the percentage of patient where we see disease shrinkage. And also, secondary endpoint includes progression-free survival. Would this delay the progression as compared to just standard Lutathera alone? So the study is now up and running at various sites. If I remember correctly, at least 14 sites throughout the US are currently enrolling the patients on this study. So please check out clinicaltrial.gov link, which is provided below this video, to see if there is a site available near you for your consideration. And we look forward to completing this study in next year and a half. And hopefully very soon after that, we can see if there is merit in testing this combination further. Thank you so much, LACNets, for giving us this platform to showcase our work and also highlight this study for the patients who might benefit from it. So please discuss this potential study with your provider, see if you will be a candidate. Thank you.